Well, it is a pleasure to be here. In fact, I've worn my Celtic tie as well, just to show solidarity with you and my friends here in Cardiff. No, it's a great pleasure to be here. We've worked quite a lot in this university, my team. And in fact, much of the data I'm going to show you were actually generated just across the road here in CUBRIC. And I won't go into why, but suffice to say, it's the best place to do these kind of experiments. As Mike has mentioned, I am a dangerous person. So this talk comes with a health advisory, and those of you who want to ennoblement from the British government, you better make sure you don't ask me any questions afterwards, because I might say things that could upset that. And of course, this is the image of me being sacked. This is the front page of the week in 2009, Silencing the Scientists. It shows the book of cannabis falling from my hand, my mouth being shut by presumably Alan Johnson. The key point about that particular caricature are the scales of justice in the bottom left-hand corner. Because there, the cartoonist has really summed up the debate. On the left-hand side, beer and fags. On the right-hand side, strange green chemicals in plastic bags. And I was arguing that the beer and fags actually are the problem we have to deal with in terms of drugs in the UK today. But the government are obsessed with what those green chemicals are. And of course, one of the problems is they don't even know what they are. So it's a kind of almost a fictitious obsession. But that debate about the relative harms of drugs was what got me sacked. And I'm going to talk you through aspects of that debate tonight. But more importantly, show you how the distortion that's currently in that debate has had a hugely deleterious effect on world science, particularly neuroscience. And that's something that we should all work to change. So you can sack me, but one thing's for sure, you can't gag me. Why was I sacked? They said I was getting involved in policy, and I said, well, as their senior advisor on policy and science, I ought to do that, you know. They said I was giving mixed messages. I said, well, horse riding is more harmful than ecstasy, which is absolutely true if you read the paper. If you go onto the web and search equine addiction syndrome, you'll find a very important comparison of the two. And that's been a remarkable paper, been downloaded over 7,000 times, my most popular paper by far. I said alcohol was more harmful than cannabis, which is true. And in fact, the government's chief scientist, he agreed with me, but not even his support could stop me from getting sacked. And I'll explain the political machinations behind the sacking in a short while. The good news is that now everyone agrees I was right, because even Obama's come out and said it. And if the American president says it's true, it is true. So I want to ask you all a question now. So I presume you've all heard of Amy Winehouse. And I want to know what you know about her death. So how many of you think she died of a drug overdose? And how many of you think she died of an alcohol overdose? And how many of you think she died of both? This is a wonderful question because you're all right. She actually died of an alcohol overdose, but of course, alcohol is a drug. And the reason that many people don't think she died of an alcohol overdose, many people do assume it was just drugs or drugs plus alcohol, but there was nothing in her body apart from alcohol at the time of death. And there's she's really the most high-profile example of how society tries to turn a blind eye to the harms of alcohol. And most people would prefer to believe that Amy died of a drug overdose or a drug plus alcohol overdose rather than alcohol alone. In fact, she's a fascinating example of the duplicity in terms of thinking about alcohol as a drug, but also in terms of the current government policy, which is focused on abstinence-based recovery. Because Amy Winehouse met all the criteria that this government sets for having been cured of her addiction to alcohol. She was abstinent and she'd been clean for at least 6 weeks. But like many people who are abstinent from their drugs of choice, she was vulnerable when she relapsed. And when she relapsed, she drank about 1 litre of vodka. Her blood alcohol level rose to 450 milligrams %, which would not normally have killed her. But because she was clean and recovered, it did kill her. And she died, as many people do, because abstinence-based recovery is fine, provided you don't relapse. As an aside, I haven't got time to talk about this today, but one of our major goals, certainly my own personal major research goal at present, is trying to find treatments to help people stay recovered, because the risk of relapse is so enormous, particularly if they relapse to heroin or alcohol. But there's no campaign. We're not seeing a campaign to tell people about the dangers of alcohol based on her death, even though it was a very high-profile one. So I want to focus a little bit on the science of pharmacology. What is a drug, and who should say what a drug is? Now, I would contend that this is what most people believe is true, that alcohol is not a drug. And in fact, a lot of the policy we have in this country today is almost complicit with that assumption, that it's okay to drink, provided you do so in moderation, because alcohol actually is not necessarily a drug. It may even be a lifestyle choice, or even some kind of offer some health benefits. But that is a belief system that has been powerfully driven by the drinks industry for centuries. This is my definition of a drug, something a politician once used but now regrets. Jackie Smith was a Home Secretary with whom I crossed swords on the horse riding and ecstasy paper. And when she became Home Secretary, they asked her the question, have you ever taken drugs? And she said, oh, I smoked cannabis, but I didn't enjoy it. And you think, well, why would you bother, Jackie? I mean, you know, it's... And she never gave an answer to that question, but I think the truth is that you had to do that to get into the Labour Party in Oxford at the time. David Cameron, when he became head of the Tory party, was asked the same question, and he said, I did things when young that I shouldn't have. We all did. Now, that we is called the Eton we, otherwise known as the Tory front bench. And the great thing is we know what they did, because some of them have told us. David hasn't, but we know from his friends what he did. And the great thing about the Conservative leaders is that they only use drugs which begin with the same letter as the Conservative Party. And there's more than one. You can work those out for yourself. So what would I say? I would say a drug is a chemical which, when taken, produces physiological changes. And in the context of the discussion tonight, we're interested in changes in the brain which are usually pleasurable and rewarding sometimes, but sometimes are damaging. And the argument about drugs and the control of drugs is about risk benefits, the harms we try to minimize, and ideally the benefits we try to maximize. And here are two examples of people who've died from taking drugs. The one on the left, I guess none of you will ever heard of, Gavin Britton. Just like Amy, he died of alcohol poisoning, but he died in a much more common way, which is in a drinking game. He was a university student at Exeter. He was in the golf team. After a golf match, he did what many people, women as well as men in university teams do. They engage in a drinking game. He lost the first round. The forfeit was to drink more, which made him more drunk, so he lost the second round, which the forfeit was to drink more. And by the end of the third round, he was dead of alcohol poisoning. But no one's heard of him, apart from those few people who read the Western Daily Press, where that picture came. On the other side, you have Leah Betts. Now, that's a real billboard. That's a photograph of a billboard of Leah Betts. Now, how many of you have not heard of Leah Betts? Okay, so you're quite young here. Right, so Leah Betts died before you were born, but she's still the iconic figure of ecstasy deaths. And Leah died on her 18th, or just after her 18th birthday. And the story is that on her birthday, she took 2 ecstasy tablets, probably about 80 milligrams, the same doses we used in the Channel 4 program, I'll tell you about later. And at the time, she was not sure what to do if she had side effects from ecstasy, and she believed wrongly that the side effects should be treated by drinking. So she drank 7 litres of water in the course of the afternoon, preparing her house for her 18th birthday party, and she died of water poisoning. that death was capitalized on by the drinks industry, who put her picture up on billboards all over the country with the word sorted. Sorted was the term at the time for people going into raves or clubs, having their pills, their e-pills, and if you had enough, you had 2 of the pills or you were sorted. And that image was put up deliberately to deter young people from taking ecstasy. But it was funded by the drinks industry. And so the only serious campaign we've ever had in this country to help people, warn people of the dangers of drugs, is actually funded by an industry which is desperate to keep people drinking and terrified that young people will switch to drugs like ecstasy, which are overall considerably less harmful than alcohol. If we look at absolute metric of harm, the number of people who die as a result of using the drug, we see that tobacco, the left-hand bar, is far and away the biggest killer in the UK today. 80,000 deaths a year. The next one is alcohol with 8,000 deaths. And then opiates about 1,200, and then paracetamol about 250, cocaine about 200, etc. And the drugs that exercise politicians to the point where they go almost hysterical, cannabis, ecstasy, mephedrone, they're almost vanishingly small in terms of the number of deaths. And you will see on Friday that there's going to be an interesting public debate on the newspapers, in the newspaper and the media, about the current government statistics on legal highs. Some of you may have heard that there were claims that there's been a massive 300% increase in deaths from legal highs in the last year. We have data, 2012. And when you look at that data and you look at it properly, you discover, in fact, that's both untrue, and the total number of deaths from legal highs is 11. So that just goes to prove the point I'm making, that there's hysteria in government about drugs. And I think, personally, that is a deliberate ploy to avoid confronting the big issues of tobacco and alcohol. And particularly for young people, the issue is alcohol. Because although tobacco kills more people, they tend to live long enough to reproduce and to die in their 50s and 60s, whereas alcohol kills right across the spectrum. Now, over the years, I worked for 10 years for the government, trying to make sense of the drug laws, trying to bring evidence to the decision-making. And as part of that process, I developed this 16-point scale for assessing the harms of drugs. And it looks at every possible harm of a drug, from whether it kills you when you first take it, or when you subsequently take it, like Amy, to whether it creates international problems like deforestation in Colombia through the cocaine trade. It is the most sophisticated set of criteria relating to drug harms, and we spent a lot of time assessing a range of different drugs on these 16 criteria, using the most advanced technique for us comparing different parameters, multi-criteria decision analysis. And from that, we came up with this figure. So this is the figure published in the Lancet in 2010, ranking the relative harms of different drugs in the UK today. This has become a very famous graph because it shows several things. It shows that alcohol is the most harmful drug in the UK today, and that cannabis is less harmful than tobacco, and that some of these other drugs which young people use, like ecstasy and mushrooms, are very low in terms of harm. But the graph is sophisticated in the sense it compares, or allows you to compare, the harms of drugs to the user, the blue bars, and the harms of drugs to society, the red bars. And you can see that the reason alcohol is the most harmful drug overall is because of the huge red bar. And of course, that red bar is driven by the fact that alcohol is so widely used. And alcohol is responsible for probably over half of all domestic violence, more than half of all child abuse, up to about 50% of road traffic accidents, a vast number of acquisitive crimes, etc., plus huge healthcare costs. So the vast social burden of alcohol makes it the most harmful drug in the UK today. To the user, the most harmful drugs are heroin, crack, and crystal meth. The next one's along. But if we need, as a society, to do something about the harms of drugs, and I would say we obviously do, because alcohol costs the UK economy about £30 billion a year, that is the drug we should be focusing our attention on. And the interest in these rarer drugs or less harmful drugs is, I think, deliberate ploy to avoid taking on the challenge of big alcohol. The alcohol industry is seen as just too difficult for governments to take on. Now, the other point that came out of this analysis was that there is absolutely no correlation between the harms of drugs and the Misuse of Drugs Act. Now, the Misuse of Drugs Act is supposedly based on the harms of drugs, but there is no correlation. And that tells us two things. It tells us the law is incorrect, and therefore it's unjust. And having 1 million young people in this country with criminal records for cannabis possession is one of the grossest injustices in the last century, in this century. It's destructive to their careers. It limits their ability to get into positions like teaching, police, medicine. And it serves absolutely no purpose other than to create an underclass. It has no health benefits whatsoever. In fact, it may have many disbenefits. But that's not what I want to talk to you about today. That's another talk. I want to focus on a topic that I have become more exercised with in recent years, which is the fact that the drug laws also have a hugely deleterious effect on research and on treatment innovation. And this effect is largely centered around the treatment of brain diseases. And that's what the rest of my talk will be about. If you're inclined to learn more about it, you can read this review, which I published last year, that goes into, in great detail, examples of how drugs like cannabis, psilocybin, LSD, MDMA, ketamine, methadone might all have, or will all almost certainly have, useful therapeutic targets that are being denied by the drug laws. And I just want to focus on one of them. I want to focus on psychedelics, because I think in many ways they're the most interesting to the neuroscientist. And I'm going to share our work on psychedelics with you today. And I want to take you back to these two Nobel Prize winners. So these are the two most important Nobel Prizes in medicine ever. On the left, Crick discovering the double helix and then going on to discover how proteins are coded by RNA. And on the right, Kary Mullis. Kary Mullis discovered the PCR reaction. That is the reaction that allows you to know how much horse meat is in your burger. That reaction has transformed not just life sciences, but also the food industry. And Kary Mullis explicitly said, I saw the PCR reaction as I took an LSD trip. I drove down the freeway in San Diego, and I saw the DNA molecules unraveling in front of my eyes as a result of LSD. And he's a keen protagonist of the use of LSD in science. Crick was different. Crick did his very clever work decoding the genetic code in the 1950s before he tried LSD. But soon after, he started using it in Cambridge, and that's why he got interested in consciousness studies. But unfortunately, the police got interested in him, and so he left. And we lost a Nobel Prize winner to Arizona because he wasn't comfortable with the idea that his lifestyle might be subject to police interrogation. It's quite amusing. He's only coming back, now he's dead, with the Crick Institute in London. And I wonder if they're going to study LSD there. I don't know, but we'll see. Mullis unquestionably would say this, that no problem can be solved from the same level of consciousness that created it. And I think one would intuitively agree with Einstein that it's not easy to solve problems if you just approach them in a stepwise fashion. And of course, his great achievement was discovering the concept of relativity through thinking really outside the box. Now, the fascinating thing about LSD was that when it was discovered, it was seen by scientists at the time as being the tool to understand the brain and to change the treatment of mental illness. The drug was made freely available to scientists around the world by Sandoz, the drug company. Such a different situation to what we have today, where it's almost impossible to get hold of any drug unless you sign away your mother-in-law, or mother probably. It was studied, there were hundreds of psychiatrists used it worldwide. There were 1,000 clinical papers, 40,000 patients were in trials. There were 40 books, 6 international conferences. And if you read this very comprehensive review by Masters and Houston, overall, when used in a sensible fashion, in a therapeutic, caring fashion, LSD was safe and, in many cases, effective. And here's an example of a meta-analysis of the 6 LSD trials for alcoholism that would have been pulled together in a modern meta-analysis very recently. There's the paper, here's the data, and you can see that the effect size of LSD psychotherapy, that's 1 or 2 sessions of LSD to help people think differently about their addiction. The effect size there is great or greater than any current therapy we have for the treatment of alcoholism. But LSD was banned in 1964, and since then, there's been only 1 clinical trial, and that trial was published last week. So in 50 years, there's only been 1 trial when there were 6 in alcoholism in the decade before that. And that is truly bizarre. Why would something with so much therapeutic opportunity be banned? Well, the truth of the matter is that LSD was banned because of the Vietnam War. And young Americans were very disaffected with the idea that they should be sent to South Asia to kill Viet Cong. And they tended to go to West America, San Francisco, and while they were there, the flower power culture developed, and people like Timothy O'Leary started developing a mindset, a cultural phenomenon of tune in and drop out. And that presented a real threat to the American way of life, which is about young men going off to fight and then come back and become senators. And they decided they had to do something about it. And they invented stories about the harms of LSD to convince at least the media that there was some reason for banning it. And I have to say, however little you think of the Sun, and the stories over methadone made me tear off my scrotum, which was one of their stories, and we did write to them and say, could you give us that case report, please, so we could verify it, and they refused, because of course it was made up. But the LSD ones are even more amazing, and I can reassure you, people do not give birth to frogs if they take LSD. But despite the fact that these are utterly ludicrous claims, there was sufficient noise to get the drug banned. And what's remarkable, it was banned in the face of opposition from the man who would be president until they killed him, Bobby Kennedy. And he asked this question of the Drug Enforcement Agency: why, if trials were worthwhile 6 months ago, why aren't they worthwhile now? We keep on going around and around. If I could get a flat answer to that question, I'd be happy. Is there a misunderstanding? So that he knew that this was not a scientific decision. This was a regulatory decision, not even a political decision. And he said, I think perhaps we've lost sight of the fact that LSD can be a very, very helpful in society if used properly. And maybe that's why they shot him. I don't know. But anyway, the drug was banned, and it has been banned for the last 50 years now. And I think that's probably the worst example of research censorship, certainly in neuroscience. And in fact, I would argue it's the worst example of research censorship in science since 1616, when the Catholic Church effectively banned the telescope and the writings of Copernicus because it didn't want people to know that the Earth went around the sun. It wanted people to believe the biblical tale that the Earth was the center of the universe. The banning of that work lasted 150 years. It's 50 years now, so we're a third of the way with LSD and other psychedelics and drugs like cannabis. Hopefully we won't exceed that record, because that was truly appalling. And in fact, when you look, as I said already, at many banned drugs, they have real potential as treatments. In fact, with the exception of crack cocaine, it's very hard to find a banned drug that doesn't have potential therapeutic utility. And here are some examples. But it's almost impossible to research them. And the fact that there has only been one trial of LSD and one scientific study on LSD in 50 years, 3 studies of psilocybin in 50 years, tells you that although you are allowed to do this research, it's almost impossible. So this is a double standard. Governments say, well, we don't stop you, but they make it so difficult that no one can do it. And beyond that, because no one's doing it, no one's interested in doing it, because people do not know, like we didn't know until we started doing it, the value of doing it. So we started doing this research simply because it seemed that we should know. It was wrong that the therapeutic potential, which had been present in the 50s and early 60s, was not being explored using modern neuroscientific methods. So this is the work I want to share with you today. It's been pump primed by the Beckley Foundation, which is a foundation in Oxford that's interested in using drugs which are illegal to help understand the brain and develop therapies. We've done 3 psilocybin experiments, and they've all been done with CUBRIC, because the truth is, CUBRIC were prepared to work with us. Other MRI centres nearer home, to my home in London, weren't prepared to do it. They said it was too controversial, that their reputation might be sullied if they worked with these illegal drugs. We've also got a depression trial starting, which I'll tell you about in a minute. We've done 1 MDMA study with Channel 4 funded. We're starting an MRI and a MEG study with cannabis. And I don't know if John Bisson's here in the audience, is he? But we've also got permission to do an MDMA trial in PTSD. We have permission for 18 months, but we can't get funding. So let me start by going through the first experiments we did using magic mushrooms, psilocybin. There's the mushrooms. I don't think they grow in Wales, actually, so don't. We know that psilocybin, the active ingredient, works like all psychedelic drugs on the 5-HT2A receptor. And we use psilocybin because it's a short-acting psychedelic. We gave it intravenously. We gave it intravenously for 2 reasons. One is we wanted to produce a step change in function, because that's gives us the most statistical power in the imaging studies we use. But also it's too expensive to use orally, because although you can go and pick some mushrooms up on the hills around Brecon, and if you want, to actually buy psilocybin from a chemical supplier that you can use orally is just too expensive. So we used intravenously, 2 milligrams intravenously. We're particularly interested in the receptor that psilocybin works on. This is the 2A receptor. You see here the human brain, the PET image of the human brain, and you'll see the hot spots. Those are the 2A receptors, and you'll see they're densely expressed throughout the cortex. They've evolved as the human cortex has evolved, but no one has any idea what they're doing. You can see where they are. This is a mouse image. You can see the 2A receptors are on the layer 5 pyramidal cells and the associated interneurons in that part of the mouse brain. And if you can see a knockout mouse, they don't have the receptors. So we know where they are, and those particular neurons are very important in integrating descending information from other parts of the cortex. So these are important receptors, we think, and we decided it would be interesting to see what happens when you stimulate them with psilocybin. So we did the experiment. It's a very simple experiment. People can distinguish pretty well whether they got psilocybin or not. You don't need statistics. Saline tends to make you feel normal, and the blue bars are the psilocybin. Even in a MRI scanner, you get very profound effects, a whole range of different effects, altered sense of perception, visual perception, time perception, etc. So a very clear effect that lasts for about 30 minutes or so. And these are the very first results. This is the ASL study looking at blood flow. And what you see there, the blue lines are the effects of psilocybin on blood flow in 3 brain regions, the 3 brain regions which are highlighted below. The red's the anterior cingulate cortex, the blue is posterior cingulate, and the green is thalamus. And you see the blue lines are lower than the grey lines, the placebo. And to our absolute surprise, psilocybin only reduced blood flow, and it did it in certain parts of the brain. Now, we had not predicted that at all. We had assumed from a literature on delirium that increased or changes in visual function, such as seeing lots of very interesting, pretty colored polygons flowing around in your visual field, would have been associated with an activation of the visual cortex, but it wasn't. In fact, there was no effect on the visual cortex at all. What psilocybin did was shut off brain activity and blood flow in those three parts of the brain. And that seems so bizarre, we thought we'd better do it again, but this time we do it using a slightly more traditional technique called fMRI, the bold response. And we got exactly the same effect. You can see the top images of the ASL and the bottom of the bold, we got almost the same effects. Psilocybin decreases brain activity in certain parts of the brain, not everywhere. The parts of the brain it decreases activity are these critical connector hubs, the anterior and the posterior connector hubs of the brain, which essentially integrate the activity of the very many different brain regions. So what psilocybin does is it actually disconnects the brain. And it allows the brain to do what it's normally doing when it's not constrained by the years of experience that have taught the brain to do what it should do. And I think when you're looking at when someone's under the influence of psilocybin and seeing a whole range of different visual images, you're actually seeing what primary and secondary visual cortex are doing. You're actually seeing the brain as it is receiving information rather than before it has been then coalesced into the predicted kind of imagery that we see all the time when we know what we're looking for, like various heads in the audience here. So psilocybin disconnects the brain. And that's, as I say, that was a remarkable finding. No one, I think, could have predicted it. We certainly didn't. And a lot of people say, well, it's just an artifact anyway. You're just changing blood flow. We tried to explain to them that's what MRI measures, but anyway. But beyond that, we thought, well, maybe we can go and look at neuronal activity. So we came back to Kubrick and we did MEG. And so that was the third study. Here's the MEG scanner across the road here. And that only looks at electrical activity of the brain. So we can get away from this artifact of blood flow argument. And again, we found regionally selective changes in the electrical power, the oscillatory power of different frequency ranges in the brain. And again, largely the posterior cingulate cortex, which is one of the key areas of the brain for determining the sense of self and the sense of awareness in time and space, profoundly affected by psilocybin, and also, to some extent, these frequencies, the anterior cingulate cortex. So we think we say very categorically now that stimulating those 2A receptors has a profound effect on the parts of the brain which integrate brain function. We could even go further because of the time resolution of MEG, working with Carl Friston and his team at UCL. We were able to estimate the likely target effects of psilocybin using this brain model, this model of a little cortical circuit with two pyramidal cells, two inhibitory neurons, and we were able to, well, I couldn't do it, but people like Suresh. Is Suresh here? You're working, you had your hair cut. God, no wonder I don't recognize you. See, you don't expect to see things you don't expect to see. It was possible to compute, based on our knowledge of how this little microcircuit works, that the major effect of psilocybin was on the deep pyramidal cells where the 5-HT2A receptors are. And I think that's the first example in human science, of being able to say a drug effect is mediated through a single class of neuron. Again, some of the referees didn't agree with us, but they couldn't come up with better examples. I also want to show you this, because we've been working with the MEG people here also to compare the effects of other kinds of drugs. And on the left-hand side, you have drugs that predominantly affect the GABA system. Tiagabine enhances GABA availability in the synapse and possibly outside the synapse through blocking reuptake. Propofol is an anesthetic, it's a GABA agonist. And then psilocybin. And, well, you don't need science to tell you that psilocybin is very different from the other two. The other two tend to increase the power of slow activity, or in the case of propofol, the beta and the low gamma activity, whereas psilocybin dampens everything down. So fundamentally different brain fingerprints of these drugs. And based on that and other thinking about how these systems work, I've sort of thrown out to you this kind of concept, simplistic concept. I'm a psychiatrist, of course, you know. I can only deal with two things, id and the ego, simultaneously. And this is how I'm thinking about the way in which those two systems, the inhibitory system, the GABA system, and the 5-HT2A system, might work differentially in the brain. So the GABA system is the vertical axis here. You increase GABA, you go to sleep. You get a lot more of this slow activity in the brain. If you decrease GABA by increasing glutamate, you stay awake and you have memories or seizures. You have too much of it. So there's an arousal component. There's an axis of whether you're awake or asleep. And then there's another component to the right here with the 5-HT2A stimulation, where 2A stimulation produces changes in valence or meaning. And the changes you get with 2A stimulation are completely accessible to memory. People remember exactly what's gone on. There's absolutely no perturbation in terms of arousal. It's just you change the way the brain works. You change the valence of the brain. In some cases, people feel better. And you just change the way in which the different cortical areas integrate. So that's the way I'm kind of thinking about what these two very important cortical systems might be doing. And we're still not entirely sure why the 2A receptors are there, but it may be they're important in maintaining wellness, well-being, and allowing people to be resilient to stress. We also know that the receptors have something to do with mood. We know that in depression and people with pessimistic thinking, there may be some upregulation of these receptors, possibly to compensate for a deficiency of serotonin. And this may help explain conditions like depression, where people get locked into a mindset which is habitually negative. And what was remarkable about our studies, particularly this is the fMRI study, was that one of the regions that was switched off by psilocybin was this region here, the the subgenual anterior cingulate. This part of the brain in the yellow circle there is a part of the brain which we know is paradoxically overactive in depression. And psilocybin switched that off. And maybe that switching off contributes to the sense of well-being that many of our volunteers got after having psilocybin. Remarkable that they're getting that even though they're having it in a scanner. And in fact, when we pull together the literature on that brain region and compare other treatments for depression with psilocybin, we see an interesting convergence. So for instance, we know that intensive meditation can lead to the switching off of that anterior cingulate region the same way as psilocybin does. So maybe that explains how mindfulness therapy can produce an improved sense of well-being. And we know that many treatments which we currently use to treat depression also switch off that brain region. SSRI, CBT, sleep deprivation, ECT, even placebo, if you get better on placebo, will switch off that part of the brain. So that may be one explanation for the therapeutic benefit of psychedelic drugs. It may switch off bits of the brain which are over-connected. And here's further evidence for that. And I just want to show you this. This is another way of looking at brain integration. Some of you will saw that my intro slide, talking about Brain Awareness Week, if you looked at the top right-hand corner, there was a brain with two little parts of the brain lit up, front and back. Well, that's this. That's the default mode network lit up. Anterior and posterior cingulate. Normally, these parts of the brain which integrate cognition and cognitive function and consciousness are working in synchrony. Under psilocybin, they become disconnected. In fact, they're out of phase. The same we found with the MEG. They were in phase and they become out of phase after psilocybin. So psilocybin really does disconnect that network we call the default mode network. The network which is a network which is you. When you're relaxing, thinking about what you're going to do tonight after you've finished listening to this boring lecture, planning what you're going to do tomorrow, the things, your internal conscious processes are run through the default mode network. Very difficult to do that sort of thinking when you're under psilocybin, by the way, so think about that. But the default mode network is over-connected in depression. Here, a very elegant French study showing considerably greater connectivity in depressed patients than in the normal controls. And that increased connectivity is associated with negative rumination. So this idea that the depressed people get locked into a mindset which is negative because they over-connect, they over-engage in negative thinking. And this reminded me very much of one of the great quotes from one of the pioneers of psychedelic research, Aldous Huxley. An interesting man, wanted to be a doctor, couldn't be a doctor because he had damage to his cornea, so he couldn't see very well, turned to literature and science, the merging of the two, took peyote cactus, wrote the book The Doors of Perception, and also brought home to people who wouldn't have read William Blake this wonderful description by Blake of how the brain works. And Blake said, if the doors of perception were cleansed, everything would appear to man as it is, infinite. For man has closed himself up till he sees all things through narrow chinks of his cavern. So Blake was a mystic who could, by virtue just of the way his brain was worked, he could see beyond the traditional way of seeing. But what Huxley said was psychedelics open up that chink in the cavern and they let people see a bigger world. And he's right. Modern theories of consciousness suggest that consciousness is something which is imposed on us by this massive learning which occurs from the day we're born. And consciousness is about seeing what you predict to see in the world rather than what is necessarily there. But the most remarkable thing about the Blake quote is this. Because if you talk to a depressed patient, what they see through the chink in their cavern, they don't see sunlight. They see the fires of hell. They get locked into a distorted thinking which potentially could be liberated by psychedelics. And we, based on our research and understanding of the potential, we got a grant from the MRC 2 years ago now to study psilocybin in resistant depression. The ethics committee made it quite difficult for us, 3 iterations, a year. The drug supply took a year, and the regulatory approvals were still 18 months on, waiting to get approval. And that is proof of how the current regulations make it almost impossible, even for people with my kind of resilience and drive, to do this kind of work. And it's ridiculous. I can give heroin in my research to heroin addicts without needing a license at all because I'm a doctor. But psilocybin is seen as so much more dangerous that I have to have a special license and have another CRB check so that I'm allowed to use a drug which I could pick on the streets of Brecon or the hills of Brecon any time I wanted. And the cost of doing this is 10 times more than to do any study with in any sort of non-illegal drug. And that brings me to the Channel 4 program. How many of you saw that? Oh, you do have TV here. That's good. Yes. Sorry. That's a Bristolian speaking, you know. That's the big brain, by the way. People say, why did you take money from a TV company to do science? And I say to them, well, because I spent 5 years trying to plead with the MRC and the Wellcome Trust to let me do a study of MDMA. Why did I want to study MDMA? Well, it's a drug that 1 million people a week use. It's been used for the last 20 years. Before it was banned, it was being used extensively in psychotherapy, which is what, as a psychiatrist, I do. It's the only drug I know that increases empathy. If I want to understand the nature of empathy in the brain, I need to study MDMA. Please, can I have some money? And the MRC said, no, it's not addiction, and we only fund addiction research. And the Wellcome Trust said, Oh my God, it's an illegal drug. Oh no, go away, never, go away. And eventually Channel 4 came to me and they said, we want to do something about outrageous on TV. Would you give people cocaine? And I said, no, actually, I'm not interested in cocaine. I know what it does in the brain, and actually, that's just TV. I want to do science. So they went away, and about a month later, they came back and they said, well, what Class A drug would you do? And I said, I'd do MDMA for the reasons I've just given you. And they said, okay. So this is a remarkable study because it's a proper scientific study with a little embellishment for television around it. So a few people that you know, Lionel Shriver, etc., being filmed, being part of the study. But the study, encased within the bigger study, there was this beautiful, balanced control group of 18 people who had MDMA, placebo, a variety of different tests. So it was a win-win. We got to do the study we wanted to do with MDMA, and they got the TV program, which is the most downloaded Channel 4 program ever. I'm particularly interested in the therapeutic uses of MDMA because of post-traumatic stress disorder. That is an amazing statistic. More American soldiers kill themselves than are killed by the enemy now. And there are several reasons for that. One is that the wars they're fighting don't make any sense to them, or anyone else for that matter. And the second is that they become very traumatized by seeing their friends and colleagues blown to pieces by an enemy they don't know. And they develop PTSD or depression, and then they kill themselves. And this is the future of war, I think. Actually, war will be more about self-harm than about harming others. But that's another story. But how can we help these soldiers who are doing what our governments tell them to do, and then coming back and being very traumatized? Well, we can treat PTSD, and treating PTSD is not easy. And this is a remarkable study that got me interested in this field by a group in the States, Mithofer and his colleagues. And they took people who had 2 failed PTSD treatments, and then they randomized them either to 2 MDMA sessions or 2 placebo sessions. And the way MDMA is given in this context is to give a reasonable dose. They gave about 125 milligrams, and which gives you an effect for about 6-7 hours. And during that day of MDMA, they worked through the trauma, and they tried to help people deal with the trauma. And we know that overcoming trauma requires the ability to relive the trauma and the memory of the trauma and use cognitive mechanisms to suppress the emotional reaction so that eventually you can talk about it without having that full-blown dissociative panic-type reaction that is so characteristic of PTSD. And this trial showed that 80% of the people given just 2 sessions of MDMA reached criteria for recovery, compared with only one of the people who had the placebo. So this was a remarkable, small but powerful pilot study, and almost all of them stayed well for up to 4 years now. So this is like a life-changing psychotherapeutic experience. And to my mind, that's the sort of thing that psychiatrists should be interested in and working on. So I wanted to try to understand what MDMA might be doing in the brain to produce such profound effects. So we did the study, and here it is. Here's the first MDMA study using ASL, and you see very similar images to what you saw with psilocybin. It's all blue. Blue is cold, blue is turning off of brain activity. So just like psilocybin, MDMA reduces brain activity. But you'll notice, those of you who know your bits of the brain, that it's a different part of the brain that's switched off by MDMA. It's not the cortical regions, because MDMA is not stimulating the 2A receptor. It's dampening down subcortical regions, particularly the amygdala and the hippocampus, where there's a lot of 1A receptors. And what we think MDMA is doing is releasing serotonin, and that's working on these inhibitory 1A receptors to dampen down the emotional reaction, and that dampening down requires a switching off of brain activity, what we see here. And during that study, we were able to look at the impact of MDMA on both positive memories at the top, as those of you who've used MDMA will know. MDMA tends to increase the positivity of most things, including memories. But more importantly, it decreased the valence, it decreased the negativity of negative memories. And that was one of the questions we were interested in. Would it do that? And it does, and that may be why it's useful in terms of a therapeutic tool. And as I said at the beginning, we've had permission from the NHS here to do a study like that in veterans here, because this is a national centre for the treatment of PTSD. And although the study is approved, it's not funded. Because I think it's very difficult to persuade people to work in this field, which is seen by many as being encouraging young people to take drugs. And obviously that's ridiculous. It's like, you wouldn't say, well, we're going to get rid of codeine because some people abuse heroin, would you? But that's effectively what's happening with these drugs like MDMA. They're because people are terrified of recreational use, there's an irrational fear of any use. And I'm going to finish, because some of you may have heard about this. It was the subject of that Science article I showed you at the beginning that makes me dangerous. I know that modern neuroscience could provide us with a safe alternative to alcohol. You could drink, become pleasantly inebriated without damaging your heart, your liver, your brain, without getting addicted, and be less likely to be aggressive. And you could even have an antidote, so that you could have a few drinks after this and then sober up and drive home safely. To my mind, that would be one of the great achievements of modern science. 3 million people a year worldwide die of alcohol-related illnesses. We could reduce that to nothing. So that's more people than die of AIDS, tuberculosis, and meningitis put together. So why don't we do that? Well, we don't do it for one reason, that no one has the confidence to progress this in case media hysteria got the safe alcohol banned. That is why no one's in this field, because no one knows if it's going to work. No one knows whether it'll be legal. Although, I can tell you, the compounds we have identified are legal. But can you guarantee they'll stay legal? You can't guarantee anything in a world where drug policy is determined by the media, particularly the Sun and the Mail. And that, to my mind, is the biggest challenge. We have to stop this current ridiculous hysteria that the media generate around drugs and try to have a rational debate how to use science to improve the health of people. And I'm completely in favor of people taking drugs if it makes them feel better. What I don't want is for them to be harmed by it. And I personally don't see why, if I have to use alcohol to intoxicate myself, as I probably do at the meal tonight if the Sackler money runs that far, why I should be killing my liver at the same time. Why? Maybe you're going to provide something else, Mike, I don't know. Anyway, what I hope is this. I think I've reached a certain status in life now where my science has become the subject of Times cartoons, and that's actually a rather clever one. But I hope perhaps in humor veritas as opposed to in vino veritas. We'll see. So I'm going to finish. I want to say thanks to all the people that did the work. I'm sorry that the Cardiff ones are the black and white. That's all they give you in this place. I mean, you never get a colour printer one day, guys. But thanks to you all for helping us do this. It's been a very interesting and stimulating ride over the last decade. And I think it's one we're going to stay riding for a while. And I'll stop and take questions and say, those of you who are interested in this field, become a friend of the Independent Scientific Committee on Drugs. Visit the website. We're the only group in the world that tells the real truth about drugs. We're free from any government support, from any industry. And if you want to know the truth about drugs and actually get the world to know the truth about drugs, please support us. Buy the book. It's cheap. All the proceeds go to the ISCD. If you buy it for your children, they'll think you're really cool. If you buy it for your parents, they'll let you read it. Thank you very much.